All right, so our discussion of nucleic acids is incomplete until we talk about manipulation of nucleic acids and techniques associated with, excuse me, lab techniques associated with nucleic acid. So, nucleic acid fragments are generally easy to work with since they can very easily be sequenced, duplicated, modified, expressed. This duplication is a result of the polymerase chain reaction, a technique developed by a guy named Carey Mullis. Carey Mullis was awarded a Nobel Prize for this in I think it was 93. Um, and he, I think to me, he's always an example of someone who is, he's really an interesting character. Obviously very intelligent, obviously brilliant, smarter than I could ever imagine. But also he has some what I would consider to be some pretty radical ideas about HIV and AIDS. He aligns himself with a guy by the name of Peter Duesberg, who basically is an HIV denialist, denying the connection between HIV and AIDS. And Carey Mullis aligns himself with, or when Carey Mullis passed away a couple of years ago. But he aligned himself with that and kind of gave that movement a little bit more notoriety because, oh wow, there's a Nobel Prize winner who believes this. So if you ever took a philosophy class, it makes me think about the philosophical fallacy of appeal to authority. Nobel Prize, brilliant, smart guy, absolutely. Carey Mullis, brilliant, brilliant, brilliant. Um, does that mean everything that he thinks is correct? By no means. At any rate, um, for manipulating DNA or nucleic acids, it's easier to get protein information from simply using DNA information rather than manipulating protein because proteins cannot quite as easily be duplicated or modified. Instead, it's far easier to change the DNA in order to get an altered protein. Um, when we are sequencing a DNA molecule, we're going to be able to cleave it into specific fragments, sequence each fragment, and then put those fragments back together. We'll talk about this same sort of technique and process when we get to protein sequence later on. But we use enzymes, or we use these molecules known as enzymes, these biological catalysts that are able to break double-stranded DNA. These are, or historically, were purified from bacteria. There are three different types. So think about this as a restriction enzyme is a biological catalyst that can break up a double-stranded piece of DNA. So the three different types require ATP and have non-specific cleavage. So they don't care about the sequence. They're just going to see a double-stranded piece of DNA. And if they have ATP, they're going to cut up that double-stranded piece of DNA. A type 2 doesn't require ATP and looks for a specific sequence. So a type 2 would be if we gave a sequence like ATGCAT. A type 2 restriction enzyme maybe will say, okay, well, if I see AT, I'm going to cut between A and T. And so this type 2, and again, I'm going to write this as a double-stranded piece of DNA. So this is a, there we go. So this is going to see A/T, or sorry, A and T, and it's going to say, oh, well, I see it right here, so I'm going to cut there. And then I see it right here, so I'm going to cut there. But what it's also going to do is look at the other strand, and it's going to say, oh, well, I see AT there. I see AT there. So I'm going to cut there. So I'm going to get that, basically that chunk out. I'm going to cut from the 3' of the A to the 5' of the T. So we have that A/T that it's going to see, and it's going to, since we're dealing with a DNA molecule, it's going to see both strands of it cut in both positions. So that's what type 2 is going to do. Type 3 is going to be the same as type 2, but it's going to require ATP. So when it requires ATP, that's almost like a way of saying will not work unless there's ATP present. Okay, so a little bit more detail on those. These are all examples of restriction enzymes. And those restriction enzymes have recognition sequences that are simple, and then there are some that are very complex. If you look at this first one at the top, ALU, this is providing us with only one strand because, again, DNA, it's a double helix. This is going to see A, G, C, T on one end, and then A, G, C, T on the other strand. ALU1 is going to look at this double stranded piece of DNA. It's going to say, I cut at the 3' end of G and the 5' end of C. So I'm going to cut right there. And then it's going to simultaneously be scanning the other strand, and it's going to cut there. Now, if you look at something, so that one's pretty clear. I like to think that that's pretty clear. The result of this is going to be A, G, T, C, and then elsewhere, C, T, G, A. Okay, so those two are going to be separated. Now, what bond is being broken here? Well, it's that 3' to 5' phosphodiester bond. Okay. Now, if we look at something like EcoR1, EcoR1, the single strand looks like this. G, A, A, T, T, C. Okay, that's one strand in the 5' to 3' direction. The other strand, G, A, T, T, C. Well, what EcoR1 is going to do is it's going to say, well, I cut after G. I cut the 3' end of G. So EcoR1 is going to be looking at both of these strands, and it's going to say, well, I cut the 3' to 5' end from G to A. So it's going to cut there. And it's going to look at this other strand, and it's going to cut there. So again, this restriction enzyme is going to break this phosphodiester linkage. Now, what's important to remember is this is a double-stranded helix. So we have all of these hydrogen bonds, 3 between G, 2 between A, 2 between A, 2 between A and T, 2 between A and T, 3 between G and C. As soon as these phosphodiester linkages here and here are broken, what that's going to do is basically destabilize this molecule. And what we're going to be left behind with is, actually, let me see if I can, let's see, draw, annotate. I draw a nice shape. I think I can. I'll draw it right down here. How about instead of red, it's white? Might make a little bit more sense. There we go. And what we're going to end up with is something that looks like this. Oh, I'm writing white, but now I'm going to write red. G, C, A, clear, nope, eraser. Oh, rats. Okay, draw, white, box. Okay, and now can I switch over to us? Okay. Oh, that's not showing up. Nope, that's not showing up. Learning technology. Okay, so we get G, C, T, T, A, A. That is one product that comes from this cut and this cut. The other product is going to be A, T, T, C, G. So another way of looking at that is when we break that bond, I'm going to select a different color. When we break this bond right here and right here, we lead to these hydrogen bonds being broken. So we're going to have this, I always kind of like visualizing it with my fingers. We have the bond between the tips of my fingers broken, and then my fingers otherwise have these hydrogen bonds, but they're going to be separating from one another. That's different from our other, when we look at ALU1. These types of bonds have different names, or these types of products have different names. The product shown right here, the product from ALU1, these are known as blunt ends. The products shown below, the resulting products from EcoR1, are sticky ends. So blunt ends versus sticky ends. One way you could describe it is, do the cut sites perfectly align with one another? Are the cut sites right on top of one another in the two different strands? If they are, you're going to end up with blunt ends. If those two cut sites are staggered, which is what we see with EcoR1, then you're going to end up with sticky ends. Clear all my drawings, annotate, clear all drawings. Done. Nope, mouse. So the type 2 restriction endonucleases that we looked at are going to end up with these two different products. Now, I never expect you to know the sequence of or the restriction sequences, because that's extreme. Um, but here what we have are examples of EcoR1 and EcoR5. EcoR1 is going to cut with the staggered system, and EcoR5 is going to cut where the two sites are directly across from one another. Now, whenever we have that, both of these restriction enzymes are directly cutting phosphodiester bonds. But EcoR1, when it cuts those phosphodiester bonds, you have this sort of collateral damage of hydrogen bonds are broken. So when you have blunt ends, only break phosphodiester bonds. Sticky ends break phosphodiester bonds and H bonds. These do not have an ATP requirement, and those restriction or those recognition sequences can be 4 base pairs, 6 base pairs, 8 base pairs, or even longer. Now, the value of all that is a technique known as gel electrophoresis, DNA gel electrophoresis, where you're able to separate DNA based on its charge. And one way to think about that is, well, every DNA molecule is going to have a negative charge, but the longer the DNA molecule, the greater the abundance of that charge. So if you think about something that's 50 base pairs versus 10 base pairs, 50 base pairs is going to have roughly 50 negatives. 10 base pairs is going to have roughly 10 negatives. So what we see with gel electrophoresis is these smaller pieces, or the smaller strands of DNA, the smaller products, are going to migrate through this gel much faster than the larger ones. So a 50 base pair one is going to be higher than a 10 base pair one. Now, restriction fragment So there was a time whenever sequencing of a piece of DNA was no trivial task. Now it is pretty much trivial. Like you can have a 600 base pair piece of DNA sequenced and that information returned to you within, I would say, a maximum of 36 hours. If you want it faster than that, you can probably do that. And sequencing a genome is probably $1,500 or maybe even less than that. Whereas, you know, 25 years ago, it was not nearly that easy. And so you had to rely on different pieces of technology. So if we look back at um, our list of restriction cut sites, you would be able to get some sequence information from cutting a piece of DNA with something like EcoR1. Because if EcoR1 did not cut a piece of DNA, that would be an indication that it did not have a sequence of GAATTC. It did not have that specific sequence. But if it did have that, you'd be able to say, aha, it has GAATTC. And so what was done for quite some time to get more and more sequence information about a gene or about a piece of DNA was to cut a piece of DNA with one enzyme, and then cut it with a second enzyme, and then cut it with two enzymes in combination. And so what we see here is just a randomly generated piece of DNA cut with the restriction enzyme HIND3. When you cut with HIND3, that 4 kb piece of DNA would be cut into one 2.8 kb piece of DNA and one 1.2 kb piece of DNA. When you cut a piece of DNA, that same identical piece of DNA with BAMH1, you would get a 1.8, a 1.3, and a 0.9 kb piece of DNA. So that would all add up to 4 kb, just like this would all add up to 4 kb. Now, for a combination of cutting these two things, when you cut with HIND3 and BAMH1 at the exact same time in combination with one another, you get a 1.8 kb piece of DNA, a 0 or a 1.0, a 0.9, and a 0.3 kb piece of DNA. And all of this information is valuable because you'd be able to take this 4 kb piece of DNA and reconstruct it. So let's for a moment imagine that we have this. We have one 4 kb piece of DNA, 0 to 4. And we cut it with HIND3. So we get a 2.8 kb and a 1.2 kb. Now, when you cut with HIND3 and BAMH1, well, now all of a sudden you get something that looks like this. That 2.8 kb piece of DNA gets cut into a 1.8 and a 1.0 and a 0.9 and a 1.8, a 1.3, and a 0.9. Well, if this is also being cut with HIND3, you get 1.8, 1.0, 0.3, and 0.9. So 1.8. So this is all because HIND3 cuts this piece of DNA one time. These two pieces are 1.8 and 2.8 and 1.2. BAMH1 is going to cut it two times because remember, this is a straight line piece of DNA. It's going to cut one time and then a second time. So you get 1, 2, and 3 products. Since we do not have any common fragment with HIND3, we know HIND3 cuts between BAMH1 and the BAMH1 cut sites because when we compare this and this with one another, you see that it's a 2.8 and 1.2. BAMH1, you don't have a single 2.8 kb piece of DNA. Instead, you have, or a 1.2, you have a 1.8, a 1.3, and a 0.9. Double cut. The only difference in BAMH1 and a double cut is you lose a 1.3 kb piece of DNA. So what this means is that that 2.8 kb piece of DNA gets cut into 1.8 and 1.0. That 1.2 kb piece of DNA gets cut into a 0.9 and a 0.3 kb piece of DNA. So it's important to kind of piece all that together. Ultimately, this is very similar to a logic puzzle, or it is a logic puzzle. It's to me, it's very similar to, I don't know if anyone ever did these in elementary school, where it was like a puzzle where you would say you would get a little bit of information, learn that Joe lives next to Sally. Sally lives next to Steve, but doesn't live next to Sandy. So, based on that, how can you organize these pieces of information? How can you organize this neighborhood for that example? In this case, you have Hind3 data, you have BAMH1 data, and you have the combination of those two. So what you would be able to get from all of this is you'd be able to say, okay, well, I know where this Hind3 position is, or Hind3 piece of DNA is. I know where this BAMH1 and this BAMH1 are. And ultimately, I can kind of reassemble this molecule or this double-stranded piece of DNA to give me this. So this is all very helpful, and it was very helpful for sequencing genes. And when you'd sequence this way, you'd ultimately be assembling a very complicated puzzle. So restriction enzymes were useful for this to construct and clone pieces of DNA. You'd be able to map these restriction sites on these pieces of DNA, and you'd be able to ultimately assume a random distribution of those four different nucleotides. So you wouldn't imagine that it's so you'd have that kind of organization. Now, remember what I said about the frequency of different cut sites. A tetranucleotide sequence will occur randomly every 256 base pairs. Because if you think about, let's think about one, two, three. Actually, let's start out with the simple. If you are randomly generating a piece of DNA, what are the chances that an A ends up in the first piece in the first position? It's 1 out of 4. What about a uh, piece of DNA that's 2 bases long? What are the chances that an A is in the first position and a T is in the second position? Well, it's 1 out of 4 and then a 1 out of 4. So this would be once every 16 base pairs, you're going to have AT. Now, what are the chances that you get ATA? It's 1 out of 4, 1 out of 4, 1 out of 4. So that would be once every 64 base pairs. What are the chances that you get, or how frequently would you find something that is ATAT, or sorry, ATAG? Well, it's 1 out of 4, 1 out of 4, 1 out of 4, 1 out of 4. So that's once every 256 base pairs. Now, the longer that recognition sequence, the less likely you are to find it in any given piece of DNA, any randomly generated piece of DNA. So if it's once every 6 base pairs, or sorry, a 6 nucleotide sequence, CCATAG, well, it's 1 out of 4, 1 out of 4. I got to erase this. So it's actually once every 4,096 base pairs. So that's ultimately what we're dealing with respect to frequency and randomness of a piece of DNA. Okay. Like I said, 6 cutters are once every 4,096 base pairs. Now, the sequencing and this restriction mapping was helpful for a number of different things. So, restriction analysis of chromosomes is extremely useful in determining inherited genetic defects and obtaining other genetic information. So, if somebody has a genetic abnormality and their or a region of their chromosome is cut with eco R1 and another person is not, well, then there's a chance that they don't have that genetic abnormality. So human chromosomes differ in sequence every 200 to 500 base pairs, creating or eliminating restriction sites. Treatment with restriction enzymes yields fragments with different lengths, or what are known as RFLPs or riflets. Mutational change resulted in the loss of a restriction site. You get something that looks a little bit like this: parent one, parent two, child one. So parent number one has a piece of DNA or a probe of DNA. Sorry, a they have a trait that's linked to a particular gene. Their DNA is cut with a specific restriction enzyme. And they see that there is they have a cut pattern that shows, okay, well, they only have this particular band, um, and they're looking for, you know what, actually, yeah. Um, so you're looking for the presence of that particular gene. Parent number two, maybe they don't have that trait. You cut with this restriction endonuclease, and you look for whether or not that gene or that restriction pattern is present, and you see that no, it's not. Okay, parent number two lacks it. Well, child number one has a copy from each of their parents. So this is a way also of looking at paternity and is this person your parent or not? Now, this is a technique that arguably is outdated. It's not completely obsolete, but it's a little bit outdated in part because, you know, and this is one of the challenges with science, in my opinion. Um, this is kind of like a cassette tape. Like it was invaluable for a time. Do we benefit from knowing anything about that technology anymore? That's kind of it's tough. Um, and it would be something catastrophic that would set us back that you would care about that or you would need to know about that. So these uh, restriction fragment length polymorphisms or RIFLIPs are a way of looking at a restriction pattern for a piece of DNA. But the reason I say this is a little bit obsolete is because of this, the polymerase chain reaction. This is a way of amplifying DNA, taking a very small amount of DNA and generating a huge amount of it. So this shows our process of doing PCR in 2 cycles. Each cycle consists of the same steps being repeated over and over again. Those steps are denaturation, hybridization, and polymerization. We have a double-stranded piece of DNA. What it's going to do is it's going to separate into 2 single-stranded pieces of DNA. Then a oligonucleotide primer that has been designed specifically to bind to these 2 individual strands of DNA will bind. So that's our hybridization. The last step is the polymerization, where an enzyme known as DNA polymerase will jump in, bind to these double-stranded regions, and begin synthesizing new strands to complete the double-stranded pieces of DNA. Now, with all that in mind, there are temperatures associated with this because what we're doing in this denaturation is we're trying to separate a otherwise very stable molecule into 2 single-stranded molecules. Those steps are denaturation, which this is going to happen at about 98 degrees Celsius. Hybridization, that's where we want a small piece of DNA to bind to each of our single strands. This is going to occur at about 55 degrees Celsius. The polymerization is where our DNA polymerase is going to be active, and that's going to take place at about 72 degrees Celsius. So this is going to be going over and over and over again. From 1 piece of DNA, you get 2. From 2 pieces of DNA, you get 4. From 4 pieces of DNA, you get 8. From 8 pieces of DNA, you get 16. From 16, and so on. So you're going to be amplifying single pieces of DNA, or you're going to be amplifying one single piece of DNA and making huge amounts of it. So cycle one is just that initial denaturation, hybridization, polymerization. Um, Stage two or cycle two is where you have that happening, but now you're not dealing with the original piece, but instead you're dealing with copies. So you're making copies of copies at this cycle two. Um, Now, ultimately, this is something that revolutionized biological research. Um, the requirements are this for this: you have to have your piece of DNA, you have to have a pair of oligonucleotide primers that will anneal in this hybridization step to your template DNA. You have to have a heat stable polymerase, a DNA polymerase that will actually do like one that will actually synthesize these new pieces of DNA, but also, maybe more importantly, a DNA polymerase that will extend and make these new pieces of DNA. So, this heat stable DNA polymerase, in addition to being able to synthesize the new piece of DNA, one of the key parts about it is that it has to be able to endure this temperature change. It has to be able to be heated up to 98 degrees and remain functional, because all of this is mixed into a microcentrifuge tube. So it's going to be heated up to 98 degrees, cooled down to 55, and then at 72, it can actually work where it wants to work. The next component is you have to have all four, this is key here, DNTPs, or our deoxynucleotide triphosphates. And that N stands for any or all of our nucleotide triphosphates. One last component that is critical for PCR is magnesium. That's a divalent cation that helps stabilize our nucleotide triphosphates. Some of the applications of PCR, well, PCR is involved in um, forensics. It's involved in um, DNA fingerprinting, identifying, like processing samples from crime scenes, which you can do the same thing for or with restriction fragment length polymorphisms, but sequencing is the technology is robust enough that it's trustworthy, that it doesn't make mistakes, and your chances of contamination are lower, or you'd be able to identify what the contaminant is. Um, Medicinal, I mean, I think everyone in this class has probably taken a PCR test, but detection of infectious bacteria and viruses. It's particularly helpful when it comes to bacteria because you can identify um, what that actual, I guess, what specific bacteria is that's causing the problem, and then you'd be able to, uh, you know, take the right antibiotics rather than something kind of arbitrarily assigned, like, oh, this should capture it. Um, at any rate, so PCR is also valuable in something known as the chain terminator procedure for DNA sequencing. So what makes this a little bit different, though, is that you're not using a DNA polymerase. Instead, you're using a fragment of a DNA polymerase. This is known as the Klenow fragment. Um, you need only one primer as opposed to in PCR where you need two primers because you're only sequencing one strand of DNA. In addition to that, you need your deoxynucleotide triphosphates, but you also need small amounts of dideoxynucleotide triphosphates. So molecules that have no OH on either the 2' and the 3' position. And here's an example of a DDNTP. So DDNTPs are relevant to DNA sequencing, but they're not a part of your traditional PCR for amplification. And the reason is because when you don't have that OH group at that 3' position or the 2' position, well, then you cannot complete this step right here of synthesizing our new molecule. So effectively what this is showing, and what is intended to be displayed here, is your DNA polymerase is grabbing, say, this DATP molecule right here. And what it's going to do is it's going to line up the A and the T. And then what it's going to do is take this phosphate group, basically make it produce a phosphodiester linkage with a 3' OH group on this DGTP. But then if we had hypothetically an H here instead of that OH, well, that would be our dideoxynucleotide triphosphate. So you would lack that, in which case you couldn't make a phosphodiester linkage. So the key takeaway for this dideoxynucleotide triphosphate is that it cannot make 3' to 5' phosphodiester linkage. All right. Well, that's going to wrap up our discussion of DNA technology, and that's going to wrap up our discussion of nucleic acids. So this is actually the conclusion of the material for your first exam. And if you have any questions about any of this material, please let me know, and I will do my best to get you an answer as soon as possible. Thanks.